Hi everyone, I am Jeraldine Latorre, and our topic for today is about mutation and gene regulation. First is mutation. Mutation is a change in a DNA sequence. It occurs when a DNA gene is damaged or changed in such a way as to alter the genetic message carried by that gene. So, mutation is the permanent alteration of the nucleotide sequence of the genome of an organism, of an virus, or extra chromosomal DNA or other genetic elements. Mutations result from errors during DNA replication or other types of damage to DNA. In mutation, there are four types. In chromosomal mutations, there are insertion, deletion, duplication, and the last one is inversion. The first one is insertion. It is the addition of one or more nucleotide base pairs into a DNA sequence. Insertion, also called an insertion mutation. This can often happen in microsatellite regions due to the DNA polymers sleeping on a chromosome level. An insertion refers to the insertion of a larger sequence into a chromosome. Next type is deletion. It is a mutation, a genetic aberration, in which a part of chromosome or a sequence of DNA is left out during DNA replication. Deletion means to delete one or more portion of chromosome. The third types of mutation is duplication. It is a type of mutation that involves the production of one or more copies of a gene or region of a chromosome. Duplication occurs when some portion of chromosome get repeated so that genes located on the repeated portion are represented more than once in that chromosome. And the last type of mutation is inversion. It occurs when a part of the chromosome breaks off, flips around, and becomes reattached. So, the order of the genes becomes reversed from what it was. A trick to remember this is that inverse sounds like reverse. Let's move in gene regulation. Gene regulation is the process of turning genes on and off. It ensures that the appropriate genes are expressed at the proper times. Gene regulation, during early development, cells begin to take on specific functions. Gene regulation can also help an organism respond to its environment. Bacterial genes are often found in operons. Genes in an operon are transcribed as a group and have a single promoter. Each operon contains regulatory DNA sequences which act as binding sites for regulatory proteins that promote or inhibit transcription. What is positive and negative gene regulation? Positive gene regulation allows the production of a gene that is needed for use at a particular times or situation. In a cell, while negative gene regulation prevents the overproduction of a gene at a particular time or situation in a cell. And where does gene regulation occur? Prokaryotic transcription and translation occurs simultaneously in the cytoplasm, and regulation occurs at the transcriptional level, while eukaryotic gene expression is regulated during transcription and RNA processing, which take place in the nucleus, and during protein translation, which take place in cytoplasm. So that would be all. Thank you and have a good day.